what is the concentration of one molecule in a bacterial cell volume. Okay, as you see, I don't like to give answers. I usually like to give questions. I think that, that's a better way to learn. And when I was giving the quote concentration, so you understand, like I'm talking about, let's say, bacterial cell, an E. coli, and let's say I have one molecule there. What is the effective concentration? Obviously, in the end, usually you don't talk about one, so it would be like 1,000,000. But then if I get an answer for one, I could just multiply and get the 1,000,000. Okay, and therefore it's good to work with one, and then you could ask, well, afterwards convert it to whatever you want. The concentration, the question is clear? Okay, so let's do a quick vote on that. Is it 1 picomolar, 1 nanomolar, 1 micromolar, 1 millimolar? Question is clear? What's below picomolar? Anybody knows? Femto. So let's add it here. Femtomolar. So I saw different ways of approaching it. I like to take something. I'm looking for usually for methods that are very robust, like that. That I know that I won't make mistakes, and that I could always use like a similar thing when I'm dealing with different problems. Okay, so I want to look at the input the data that I have. What do I know? I know that my concentration is equal to 1 molecule per 1 cell volume. Okay, that's not too difficult. It's just stating the question as an equation. Strange equation, but still something you can work with. And from here onward, I'll be using, if you want, like a tool or a trick that I'm always allowed to do, and that is multiply by 1. Okay, I'll multiply by my way by 1 until I get the answer that I want. Where the answer that I want would be somewhere here. It's supposed to be molar in terms of units. Okay, my problem is that this is not in the units that I'm interested in. It's not molar. Okay, anybody remembers what's the definition of a molar? Mole per liter, right? Okay, good. So now my only problem in life is that I'm in molecules and I want to get to moles, and I'm with cell volume, I want to get to liter. Okay, so let's start multiplying by 1. So for example, I want to get rid of the cell volume. So I can again multiply by 1. What would I can say 1 cell volume is equal to, so what is the cell volume? I remember my cell is like a micron in length scale. Okay, so it's a micron by a micron by a micron. Okay, so it's 1 cell volume is 1 micrometer cube. Okay, you see I multiplied by 1. The nice thing is that the units here are equal. So now I can say, I'm doing it the long way. I know some of you can do it more faster, but allow me to do it step by step. So this is going to fall with this. And now I have 1 molecule per 1 micrometer cube. Okay. Let's multiply again by 1. What multiplication by 1 would I do now? Let's say I want to get rid of the 1 micrometer cube because I want to get to liters. So 1 micrometer cube, how many liters is that? So I don't know, I'm getting confused with these things. 15, 18, 21, what's happening there? I don't know. But I know that micron, I do remember, it's 10 to the minus 6. And therefore, it's easy for me to say this is like 10 to the minus 18 meters cubed. Okay, because multiplying 6 by 3, that's something I know how to do well. It's 18. Okay, and that's again nice because now I'm going to get rid of this. But that's not good enough. I'm in meters cubed, and I want to get in the end to liters. So what would I do? I would multiply by 1 once again. I want to get meter cubed, 1 meter cubed. And now I want it to be liters. And that I remember, that's 1,000 liters in a meter cubed. I'm going to run into space problems, but not to worry, it would be okay. Okay, so now you see that I got rid of this again. Meter cubed and meter cubed. No, yeah, sorry, yeah. It's, okay, so far, I could completely, what I wanted is not to delete the value. I'm just deleting the units. Okay, the units are going to cancel. The values stay okay. Until now it was 1, so nothing to worry about. But here I don't want to cancel the minus 18. Okay, thanks for explaining to me what I'm doing. Okay, so it's just this. Okay, now we have the liters. Now I have the issue with the molecules versus moles. Right, so let's multiply again by 1. How do I do this? I want to get rid of the molecule, so I want to have 1 molecule here. And I want to have something with the numbers of molecules. Yeah. Or you know what? Let's do it even more nicely. You could say, I don't even remember, like how to do the minus. Let's just say 1 Avogadro number. 1 mole. And then what would be the number here? How many molecules? 6 something. We said 10 to the 24. Instead of 6, 10 to the 23, we just, order of magnitude is 10 to the 24. And you can say this was, again, what was important, it's important the units, I want to multiplying by 1, right? This is equal to this. I want to get to molecules to get rid of the molecules. And I could have done it 1 molecule, 10 to the minus 24 mole, but that's a bit less intuitive. Am I correct? Am I mistaken? Whereas just 1 mole and 10 to the 24, no way to get it wrong. Okay, and now I think now we're ready. Why am I ready? Because the molecule in terms of units would fall with the molecule, and I'm just left with moles per liter. And now I just need to see what I have. 1, 1, 1, 1, 1. Okay, that's easy. I have 1 molar divided by 1, 1, minus 18 plus 3 plus 24. What is it? Liters. Okay? Good. I hope I'm not mistaken because I can't follow on this by head. Okay, so this is minus 18 plus 3, minus 15. Minus 15 plus 24, 9. Good. 1 molar in 10 to the 9 liters. That's equal to 10 to the minus molar in 1 liter. Yes, small, sorry. So the mistake was here. Which is 1 nanomolar. We did it right. Okay, but usually it doesn't take that much energy. And the important thing is that every step here is like close to trivial. And with experience, you might do 2 such things together, 3 such things together. But the important thing is it's pretty robust in terms of like there was nothing. I didn't to have any imagination, special imagination here. And that's a useful way to do things. The sweet thing is that now, for many other things, you can now try to apply this once you have this rule of thumb. Anybody has a a suggestion of how they might apply it for something else? Yeah, concentration of a drug or anything else. Yeah. Anybody measuring something in the lab that they could use it for? So usually if you have, now you could also use it in the other way around. Like if you have a concentration of something, you can infer what's the number of molecules that you have. Okay, so I think, try to keep it in your hand next time you do an experiment and you have a concentration, and you can now easily convert into an absolute number.